What's up, everybody? Welcome to Omnipotent Health and Fitness, episode number 8. Our guest today is a professor of biology at Boston College and received his PhD in genetics and biochemistry from the University of Illinois. He served with distinction in the United States Army's 1st Cavalry Division in the Vietnam War prior to completing a postdoctoral fellowship in the Department of Neurology at Yale University School of Medicine. He subsequently served on the faculty as an assistant professor in neurology before continuing his research into the ketogenic diet and cancer metabolism at Boston College. So please help me in welcoming the man with over 180 peer-reviewed publications and the author of Cancer as Metabolic Disease, Professor Thomas Seyfried. Professor Seyfried, welcome to the show.
Oh, thank you very much. It's a real pleasure to be here today.
Yeah, thank you for taking the time to come. So I always like to start with everybody's sort of origin story. How did you get into researching the ketogenic diet and cancer metabolism?
Well, when I was on the faculty at Yale in the Department of Neurology, epilepsy was the dominant area of research and clinical focus in that department, with Gilbert Glasser as the chair at that time. And having come from biochemistry and genetics at Illinois, I was encouraged to do as much work in epilepsy as you possibly could. And I started to map genes and seizure models and these kinds of things. I wasn't really, in fact, I wrote a small grant for ketogenic diet for the Yale Medical School. And they said no one is interested in ketogenic diets at Yale University. So I said, okay. And then when I came to Boston College, we were doing genetics of epilepsy quite extensively. Published a lot of papers on the genetics of audiogenic seizures, different kinds of epilepsy models, the EL epilepsy model. One of my students said to me, Mariana Tetrodova, she said, oh, there's a big thing out in Seattle on ketogenic diets. And I wrote, they said they have monies to support me to go there. And I said, no, don't waste your time. Nobody's interested in ketogenic diets from the work I had at Yale, right? So she said, oh, no. So Jim Abrams, who had started the Charlie Foundation and was the movie producer of different Hollywood movies, and his friend Meryl Streep, they were doing first Do No Harm, the movie, and then they we're starting to gain a research interest in ketogenic diets. So Mariana had come back to me saying, we really have to start doing this ketogenic diet stuff again, despite my reluctance. And then we started to find out that it really worked in stopping the epileptic seizures in the EL mouse. And then we found out the same thing, though, that if you calorie restrict, you can get the same results. So we were publishing on epilepsy. It did a lot of work on epilepsy. Then I started to get really deep into the mechanisms of how the ketogenic diet might stop epileptic seizures. I was consulting with George Cahill, who is the president of the Joslin Diabetes Center there in Boston, and Richard Veach. So I started to talk to these two folks. They were the world leaders on understanding how ketones were operating to influence brain function and different biochemistry of many different systems. And I started to learn from Veach and Cahill the power of what these ketones were doing. And at the same time, we were investigating angiogenesis in cancer because I developed some of the best animal models for brain cancer that exist, that I developed at Yale University to study gangliosides in brain cancer. So one thing started to come together. Let's try, what happens if we do a ketogenic diet on a mouse with brain tumors? And we actually started first with calorie restriction because it was working so well against the epileptic seizures. And the mechanism was simply related to low glucose elevated ketones. So as soon as we started to do that with the brain tumors, we noticed our dramatic finding. And then there was a drug company that came to us, because I was working a lot on gangliosides, and this drug company said, "Oh, we have a drug that stops ganglioside biosynthesis." So I tried it on the mice that had brain tumors, and lo and behold, this drug that was supposed to stop ganglioside biosynthesis also significantly shrunk the brain tumors. So the company got really excited. They gave me like $250,000 to investigate this drug which we
A
didn't
lot-
know. So
A lot more money in
Oh.
that rather than Tay-Sachs disease or
Oh
something.
yeah, yeah. So anyway, listen to this. It turns out we gave the drug to the mice, and they were eating, and we saw them eating, but their body weight was getting lower, and the tumors were shrinking. And then this Dr. Mukherjee came in, who does a lot of work on calorie restriction. She says, why don't we set up an experiment where we match the body weight with the drug? Just take another mouse, don't feed him as much, and just see match. And the tumors went to the exact same size. So we said, what is this drug doing? And we found out the drug blocks sucrases in the stomach, so the animal can't digest his food. So the drug was causing calorie restriction. So as soon as I told the company that the mechanism of action wasn't through ganglioside biosynthesis, but calorie restriction, they didn't want any part of this anymore. Pulled the plug on the research, and that was the last I heard from them. So they were not excited at all to know that their drug was operational primarily by lowering blood sugars.
Yeah, that's fascinating. I definitely want to circle back on a few points, but to that point, many chemotherapeutics cause things like vomiting and GI distress. Do you think that there is a confounding factor of a little bit of caloric restriction with the chemotherapy agents that are out today?
Yeah, I do. But it's very hard to design an experiment to test that. But nausea, vomiting, diarrhea, all of these fatigue, all of these adverse effects of chemo are all different kinds of calorie restrictions.
Yeah.
And I've spoken to some people that contact me who have brain tumors, and they were taking temozolomide, which is a standard drug for brain cancer. And this one person got so sick, he just laid in bed for a whole week, just drank water. And he noticed a tremendous, he felt really healthy after with the temozolomide. But he said it shrunk the tumor. So because he wasn't eating. I mean, he's taking temozolomide, got so sick he never ate. So the bottom line is he's getting calorie restriction because the drug made him sick. So I said, what do you think would have happened if you didn't take the temozolomide and just did oh no, doctor said I take the temozolomide. But yeah, it's temozolomide, but it's not only TMZ, it's a lot of different drugs. And the big
bevacizumab, right?
Yeah, they have a bunch of them.
Okay.
Well, bevacizumab is bad.
Okay.
Okay. Bevacizumab is an anti-angiogenesis drug. Bevacizumab is Avastin.
Oh, that's
and
right.
it's made to block blood vessels. Now, I said it's unethical, in fact, even immoral to use Avastin on people, because what happens is it makes the tumor look good on a scan, but forces the cells to invade throughout the whole cortex. And there's been several papers published by different groups. So the patient looks at the image and says, "Oh yeah, it looks like the Avastin is working," and what it's doing is simply facilitating tumor division. So it's almost like sign your death certificate after you take Avastin, because it's the worst thing. And they stopped it for breast cancer because it was causing more harm than it was doing good, but they continue to use it for brain cancer for reasons I don't know, because I've looked at all the trials. It doesn't do anything. So, and it also causes the tumor cells to spread through your brain. And this has been published in the scientific literature by a number of groups. So why would you want to use any drug on a person to spread their tumor cells through the brain? Doesn't make sense, right?
Right.
All right, well, they do it. And I called them out on it, but they continue to do it. It generates a lot of revenue. It's a very good drug for revenue generation, but it doesn't do anything to help the patients.
Yeah.
Every now and then you find a person that can survive Avastin, but it's usually a pretty devastating drug. So, but that's a different. The one with Martha Stewart, right? She went to jail for a while from Imclone, something like this. So I checked into that drug, and that worked through calorie restriction as well. They notified her in advance that the drug wasn't working as well as it should, so she pulled the stock out of them. Everything here is revenue generation. You either got to make money or protect what money you have, right?
Right.
So, you go back and look at the actual paper in clinical cancer research on what that drug was. I can't remember. I can't remember the name of the drug right now, but it was the one that Martha Stewart had to go to jail for. So, um, when you go back and when you know how to look, when you know how to evaluate the data, you can kind of predict what the drug is doing.
Right.
So yes, the answer to your question is probably a lot of these cancer drugs will have some therapeutic benefit by making the patient sick and not eating, and therefore you're going to get some benefit from the fact that you're not eating.
Right. So before we jump right into cancer metabolism, I want to circle back on something. I find it interesting that both you and Dom actually started with epilepsy research. And when we were talking to Dom, he mentioned that there are cases where people go on the ketogenic diet for their epilepsy, and then they get off the ketogenic diet, and then the epilepsy never returns. And I was wondering whether you had a hypothesis for why that happens.
We don't. And, here's the situation. Studying ketogenic diets with epilepsy will be an exciting but never-ending story. I think that what you just mentioned, like how is it possible that a person could be cured of their epilepsy? It doesn't happen to everyone for sure, but it certainly happens to enough that it's been noted in the publications. But the mechanism by which that happens is not known, and nor do we really understand the mechanism by which a ketogenic diet stops epilepsy. And the reason why I get excited about it for epilepsy, because everybody was focusing on the genes for epilepsy, and it turned out that a ketogenic diet could manage to some extent a broad range of epileptic seizures, regardless of what the etiological origin of the seizures happened to be. So it was some really general mechanism that just lowered the sensitivity, the excitability of neurons in some way. And I know a number of investigators that will be continuing to study this, but I think there's so many different mechanisms that could come to play in trying to determine how a ketogenic diet might stop this form of epilepsy or that form of epilepsy. And therefore, research will be continuing in that field greatly. For cancer, it's so much more clear on how ketogenic diets, calorie restriction, stop seizures. The mechanisms are very clear to understand, and it also works against all different kinds of cancers. Yeah. So, but it's very clear to me. The problem is you have to convince the field of a very simple explanation for how it works, and that seems to be a bigger challenge than actually knowing how it works.
Right. So let's get into that. I think a good place to start would just be with Dr. Otto Warburg and talking about, his work, and then the Warburg effect, essentially, and then how that relates to cancer metabolism for the people that are hearing this for the first time.
Yeah, well, Otto Warburg was absolutely correct in his discovery for the origin of cancer. It is a chronic damage to the oxidative phosphorylation system, forcing the cell into a fermentation metabolism. How the damage to the mitochondria can come about is we call the oncogenic paradox. And then some people could get cancer from viral infections like hepatitis C, and some people can put it. These are all risk factors, by the way, risk factors that would disrupt oxidative phosphorylation, forcing the cell into a fermentation mechanism. So you can see intermittent hypoxia, chronic inflammation, exposure to carcinogens, even rare inherited mutations that affect the function of the mitochondria. All of these are risk factors, and that was what Albert Szent-Györgyi found to be what he called the oncogenic paradox. And in Sid Mukherjee's book, The Emperor of All Maladies, he also struggled to try to understand this paradox. How is it possible that so many things in the environment or chemicals or whatever could cause cancer through a common pathophysiological mechanism? And that common pathophysiological mechanism is the gradual replacement of oxidative phosphorylation with substrate level phosphorylation, which is the fermentation mechanism. The problem today is most people consider Warburg wrong because many of these cancer cells take in oxygen. And the problem there is which they use the seahorse instrument and a variety of other instruments. It's important, and I published this over and I tell people this, oxygen consumption does not equal oxidative phosphorylation in cancer cells. In cancer cells, they're disconnected. So, and this is what confuses so many people. They've already made a decision, oh, Warburg has to be wrong because the cancer cell is taking in oxygen. It's sometimes equal to that of a normal cell. Wrong. They're taking in oxygen, it's not connected to ATP synthesis. We now show, we have evidence that the oxygen, the ATP coming out of the mitochondria in the cancer cell is coming from mitochondrial substrate level phosphorylation.
Right.
So this is Warburg did not know about this. Warburg did not know this stuff came after his major contributions to the field. So what led to tremendous confusion was that cancer cells could continue to produce ATP linked or in association with oxygen consumption. But we're showing that the oxygen consumption is not the mechanism responsible for ATP synthesis. Oxygen consumption produces reactive oxygen species. It doesn't produce ATP. So the ATP is actually coming from glutamine through the glutaminolysis pathway, producing large amounts of ATP in substrate level phosphorylation. So, so he didn't know about that. And that was, we now have defined the missing link in Warburg's theory. He's absolutely correct. Origin of cancer is chronic damage to oxidative phosphorylation with compensatory fermentation. And the fermentation is the glycolytic pathway as well as the glutaminolysis pathway, both providing most of the ATP from our studies is coming from the mitochondrial substrate level phosphorylation. The glucose carbons are used predominantly for biomass synthesis, as several others in the field have shown. So the energy does not come much from glycolysis, mainly because of the PKM2 isoform. So you produce a lot of lactic acid with very little ATP. So what is all this glucose being? All the glucose is doing is largely for biomass. Without biomass, you can't grow. And without energy, you definitely cannot grow. But if you have energy and biomass, you're going to grow like crazy. And the two fuels driving the cancer is clearly glucose and glutamine. So you can clearly manage cancer by depriving cancer cells of glucose and glutamine simultaneously. Right. Not difficult.
So if I could just take a step back, it sounds like for anybody hearing this for the first time, essentially with cellular respiration, normally in the presence of oxygen, you undergo aerobic respiration, which tends to occur in the mitochondria through the electron transport chain. But you're saying in cancer cells, what happens is even in the presence of oxygen, the process for which they form ATP would be anaerobic pathways or fermentation, being glycolysis or through substrate level phosphorylation. Is that?
Yeah, that's correct.
Okay.
And what happens now is you only use a small part of the TCA cycle. So the TCA is no longer a cycle. These are pathways that so you have glutamine metabolized to glutamate, metabolized to alpha ketoglutarate, metabolized to succinyl-CoA, and then to succinate. And that step there, the succinyl-CoA to succinate, is generating the majority of ATP in the cancer cell. And as the result of this, they dump out succinic acid outside the cell. So, and a number of investigators have shown that succinic acid is accumulating in various cancer cells. And succinate stabilizes HIF-1 alpha, which then allows the glycolytic pathway to proceed at full throttle. So the two pathways for biomass and energy are tightly linked through their metabolites. And we know this. Listen, we have cancer cells now. Everybody knows cancer cells can grow in hypoxia. I mean, really low levels of oxygen, 0.1%, and they can grow in cyanide. Okay, so we have our cancer cells growing in cyanide and hypoxia, growing fine. As long as they have the glucose and the glutamine, they can grow without oxygen. So you tell me any cell, a normal cell that can survive in cyanide, right? How is a cancer cell surviving in cyanide if they have normal oxidative phosphorylation, right? They don't. So they don't use oxphos. They use fermentation mechanisms. And the fermentation mechanisms are very simple. It's glucose and glutamine. And if you target the glucose and glutamine simultaneously, you're going to get spectacular results in managing cancer.
Right. So I was wondering, I think there's a gap in my knowledge that I'm trying to understand. So with cancer cells, when they're shunting towards aerobic fermentation, essentially, what then is the steps that causes cancer from there? Is it just decreased amount of ATP? Is it increased lactate levels and causing an acidotic state? And how does that then relate to essentially causing DNA damage to cause mutations?
So, yeah, well, you have several concepts overlapping in your question. So the DNA damage comes largely from the ROS that are produced in the cell. ROS are carcinogenic and mutagenic. There's large numbers of papers in the scientific literature showing that. So the DNA damage is downstream effects. It's not the cause, the effect. It's an effect. And the nuclear transfer, mitochondrial transfer experiments pretty much solidify that.
Right. Now, actually, do you want to go into that really quickly? I
think it's important to know, okay, that's great, DNA damage. So the key thing is, what is responsible for the dysregulated cell growth? You know, this is another interesting thing before we go into the nuclear transfer experiments.
Sure.
But okay, great, mitochondria damage, how's that going to lead to dysregulation? Because the signature phenotype of cancer is cell division out of control. Dysregulated cell growth. How do you get dysregulated cell growth from damage to the mitochondria, right? Mm-hmm. So you have to realize that all of the cells that existed on the planet before oxygen came into the atmosphere were all dysregulated in their growth. There was unbridled proliferation. And they were all using a fermentation mechanism because there was no oxygen yet. So when oxygen came into the atmosphere, it's only then that you began to produce ROS as signaling molecules that allowed metazoans to form. So the differentiated state of our cells is controlled largely by the mitochondria. The mitochondria also control the genes that regulate the cell cycle. So what happens when the mitochondria become no longer operational within the cell? They fall back on their ancient pathway of fermentation, which is linked to dysregulated cell growth. So all this stuff makes real sense if you understand evolutionary biology. What I find in my discussions with students or with physicians, wherever, most people don't even understand evolutionary biology. It's actually like a black, it's very hard to convince people of mechanisms when they have no clue about evolution. And believe me, when you see people writing these different kinds of things, you already know they're using teleological explanations, using all this kind of stuff. So once you understand evolutionary biology, you understand the function of these different organelles and how they all came to pass, you can put this whole puzzle together with not too much difficulty. But the nuclear transfer experiments were clearly showing the beauty of these experiments. They were asking, what do gene mutations, how can gene mutations regulate development? But the evidence that they got was that they were actually destroying the somatic mutation theory of cancer by the very observations that they were making in transferring tumor nuclei into normal cytoplasm and normal nuclei into tumor cytoplasm. Doing frog work, mouse work, different cancer work, all doing different model systems, and majority of these people coming to a similar conclusion that there's something in the cytoplasm that's playing a bigger role in driving dysregulated cell growth than there was in the nucleus. And when you look at that, I had long discussions with Dr. Kimmel, who did the frog work. He even told me, he showed me pictures of frogs that were developed from the nucleus of a kidney tumor. And he said, frogs can regenerate their tail and limbs and things. And he would cut the tail off this tadpole that was generated from a cancer nucleus. And the tadpole completely regrew the tail exactly as it should have been, despite the fact that this is dysregulated. This is regulated cell growth from the nucleus of a dysregulated cell. Can you believe this? So clearly it was all in the cytoplasm. Wasn't it? The genes were not controlling the regulation of the growth. So when I told him it's the mitochondria, he was like really excited. He said, yes. He says, yes, you're absolutely correct. It is the mitochondria that doing all this. So that's a hard pill to swallow because you realize most of the stuff in cancer is based on the somatic mutation theory. And if the somatic mutation theory is incorrect, what is the probability of developing new drugs that are going to be really effective if the very foundation upon which you're building these drugs is wrong? It's not going to work. So simple. This whole thing is like incredibly simple, and yet people can't understand it. I don't know. I find myself, sometimes I question my own sanity. So am I going crazy here?
Right. So just to reiterate for the audience, so they did studies where they took the nucleus, which is where your genes are held, of a cancer cell, and they put it into a healthy, normal cytoplasm. And essentially that cell never formed into cancer, but they then did the opposite, where they took essentially cancer cytoplasm and then transferred a normal nucleus into the cancer cytoplasm, and then it developed into cancer. And that's kind of what the nuclear transfer experiments were, right?
Yeah, mitochondria too.
Yeah.
If you take an indolent, slow-growing tumor, and then you put in the mitochondria from a rapidly growing tumor, the slow-growing tumor becomes extremely rapid. And if you take a rapidly growing tumor, this was all done by the group at Baylor College of Medicine, Benny Kirikpatu and others. If you put normal mitochondria into the side, replace them. If these are hard experiments to do, mitochondrial replacement experiments are much more difficult than nuclear transfer experiments. So if you put the normal mitochondria into the cytoplasm of a very aggressive tumor, you can shut it down. It becomes growth regulated again. So when you put all these things together, it's clear the mitochondria are calling the shots in this disease. Cancer is a mitochondrial metabolic disease unambiguously. And once you realize that, then the strategies for managing the disease become so simple and easy, non-toxic. So, right. And that's the hardest pill for people to swallow, that this cancer is not as complicated as many people make it out to be. Right. And the other thing too is all the cancers that I've looked at, all the major cancers, they all have damage to the structure of the mitochondria. So they're all fermenting, and they're all sucking down glucose, and they're all sucking down glutamine, and there's no other amino acids that can replace glutamine. So in the same robust level, and then you also have to look at the logistics of this. What is the amino acid most abundant in the body? It's the glutamine, not these other amino acids. They're very low. So in order to grow the beast, you have to have an inventory of fuels, and the biggest fuel is glucose and glutamine, and they're both being fermented, and the cancer is growing. And nobody's targeting glucose and glutamine in any of the clinical trials. So what do you expect?
Right. So let's go ahead and talk about that. Let's talk about the press pulse protocol that you guys have been developing. And maybe you could talk us through briefly the mechanism of how a ketogenic diet and caloric restriction fits into press pulse.
Yeah, so the press pulse, I developed that from the concepts of Aryans and one of the authors, for the extermination of species during the Earth's evolutionary history. It turns out that chronic stress will eliminate the weak. A pulse stress will eliminate many, but not all organisms. It's only when you have a chronic stress together with a catastrophic pulse that you cause extinctions. And we developed that concept for how we destroy cancer, because you can't go into the targeting tumor cells like they do in the clinic with a toxic drug radiation. This will not work for many people. So what you need to, and you find out that many of the cancer patients suffer from all kinds of other maladies. Sometimes it could be type 2 diabetes, high blood pressure, hypertension. Many folks that have cancer have many other maladies, not just the fact that they have the tumor. So we look at a whole profile and we say, okay, this person needs to be brought back into metabolic homeostasis. So by doing that, we either use low carbohydrate diets, ketogenic, depending on the health status of the patient when they first appear. So the press part of this is to lower the sugar, elevate the ketones, to bring the patient back into a more healthy state than they were when they first presented. Also, by lowering glucose and elevating ketones, we put metabolic pressure onto the tumors. They don't grow as fast. Microenvironmental acidification goes down. There's a lot of anti-inflammatory benefits from this, and the body starts to get healthier and healthier. Now we launch the pulses, which are strategies that will attack and destroy the tumor cells gradually. So we can use diet drug cocktails that will target and hit glutamine-dependent cells, glucose-dependent cells. You put the patients in hyperbaric oxygen, which will cause us oxidative stress more in the tumor cells than in the normal cells, just like radiation. But radiation doesn't discriminate between tumor cell and normal cell. The hyperbaric oxygen, while under nutritional ketosis, will largely destroy cancer cells, especially when the glucose levels are reduced, just like Doug Spitz and his group showing out in Iowa. You lower glucose, oxidative stress goes up. Put a patient in hyperbaric oxygen, the oxidative stress is going to be even greater. The ketones are reducing the stress in the normal cells. It's a beautiful, elegant, non-toxic system for eliminating tumor cells. It's a gradual process. It requires participation on the part of the patient. They have to be somewhat compliant with these things. And it also requires people to understand what the hell is going on with this strategy. You just don't sit your ass down there and get nuked and poisoned. You have to know what you're doing, right? So, and the attending physicians need to know this. It's a mitochondrial metabolic disease dependent on glucose and glutamine, and they can't burn ketones. So you put all that together and you have a strategy, a press pulse strategy for managing the majority of cancers in patients.
If I could just talk about the press portion for a moment, is there any difference, or would you recommend using exogenous ketones during the press portion, or does it have to be all endogenously made?
No, you can use ketones, exogenous ketone supplements.
Okay.
The question is, do you want to use racemic ketone DL forms of mixture, or do you want to use the D form? The D form is ultimately the best. The D form will be the form that we produce naturally in the liver is D beta hydroxybutyrate. That molecule is different from L beta hydroxybutyrate. The L is beta oxidized, and then you don't get the energetic advantage from the L form as you do from the D form. It's a sophisticated biochemistry that Richard Veach, Dr. Veach, shared with me. He's published it. It's a little detailed, but bottom line, it's the natural ketone body. If you can take a solution of natural ketone bodies, the D beta hydroxybutyrate, while in a low carbohydrate diet, you will definitely elevate your ketones to a higher level than you would if you were just on the diet by itself. There's no question about that.
For those who aren't aware, the D and the L are enantiomers, meaning that they're mirror images of each other, kind of like your left and right hands, is what we're talking about with the D and L. So I was also wondering, how low is the glucose supposed to go for optimal press?
Well, it's the lower the better. And of course, the study done by Drenick in the 70s, where he took the obese people and put them on a water-only fast for 21 days, and then shot them up with high-dose insulin. They were able to get blood sugars down to 0.5 millimolar, 9 milligrams per deciliter, without any cognitive decline at all. Wow. And this is what Cahill was always telling me. He says, if you can get your ketones up, you can push glucose down to barely detectable levels. So, but otherwise it would kill the patient. But the ketones replace glucose for the brain. So the brain is perfectly capable of burning ketones as an alternative fuel for glucose. And if that's the case, you really don't need glucose. So, and that's why these people were getting, so our group in Turkey puts patients on ketogenic diets, ketone supplements, and then does insulin potentiation studies, things like this. The question is, here's the situation with press pulse. We have not yet perfected dosage, timing, and scheduling for the different parts of the process. I'm working with groups of physicians in various clinics to work, and in our lab ourselves, diet drug cocktails using dosage, timing, and scheduling. What is the best level of glucose and ketones that will make the drugs work best? Does this drug work better with this other drug or with the diet in between? So it's mostly, we call it unsexy science. You can't get funded for this stuff, right? This is ultimately what's going to solve the problem, and you can't get money for it. You have to integrate all these different pathways. Screw the goddamn pathways. You're trying to keep the patient alive in a quality of life. What are you worried about all these signaling pathways? We have to know that this will solve the problem. And for whatever reason, that end goal is not considered important. dosage timing and scheduling is not sexy science that will ultimately lead to the resolution and long-term management of the disease. But it's
Right.
trial and error, and every person's a little different from somebody else. So you might have to tweak the diet drug cocktail a little bit different for one person for another. And we can work all that out. It just takes time.
Yeah, I was wanting to touch on timing actually, and I was wondering how long you had to have been on in ketosis for, because speaking to Dom, it sounds like there's about 3 months before you're fully keto adapted. Does that play any role as far as the press pulse protocol?
No, I don't think it's that long. We're seeing better now. I think what we do is we put people on low carb, zero carb diets. You can do, you can eat whatever you want, just don't take in any carbs. And then what happens is they within a 14-day period, you can start to see the ketones going up and the glucose going down. It doesn't go down as much, but then you step into a water-only fast at that point. It becomes a much less stressful thing. And then once you see that, and then you can take the exogenous ketones. When you're in that ketogenic state, that's when you bring the drugs on. And the drugs are delivered through at least to the brain, in 3 times more passes through the blood-brain. It's unbelievable. These diets are acting like facilitators of drug delivery to the brain. So we published 2 papers on this, showing how the ketogenic diets can facilitate delivery of small molecules through the blood-brain barrier. So what are you wasting all your time trying to figure out drugs, how to bust the blood-brain barrier when you already have it here? And then when in ketosis with these drugs, these tumor cells can't survive. They're being really hammered big time, and your normal cells are not being harmed at all by this.
So it sounds like because the patient has become keto adapted, when you do the pulse and you block the glutamine, the healthy cells, the non-tumor cells, are essentially protected against the pulse because they're able to use ketones, whereas cancer cells are not.
Yes.
Wow.
Yes. Is that it's not that complicated, right?
Well, when you put it that way, but
no. I'm telling you, what I just told you cannot be accepted by the majority of people in the oncology field. They either don't know about it, they don't want to know about it, or they think, oh, it has to be more complicated than that. Oh, you tell me how. I don't know.
Yeah. So. And how does the glucose ketone index play into that? I know you mentioned timing and dosing. Is that still the 1-to-1? Is that still what we're aiming for?
It's simply a quantitative gauge to let someone know whether they really are in a ketotic state, because you're actually quantifying the level of the millimolar level of ketone and millimolar level of glucose in the individual's blood at a given time. And Dom and I know that for hyperbaric oxygen as another pulse, we like to have the patient in during the course of a day. It waxes and wanes as your GKI, because we've done this in a number of people, and you can get a different GKI in the morning than you can get in the late afternoon, or vice versa. It can change during the course of a day. So we always tell people, if they want to do hyperbaric oxygen along with their as part of the press pulse, they should go into the chamber at the time of the day when they have the lowest GKI, because that tells you now that you're going to put the maximum oxidative stress on the tumor cells. And that has to be determined for each individual patient themselves. So patient has to say over a course of a week or 2, this is when I notice that my GKI during the course of the day is the lowest. So you schedule your hyperbaric oxygen during that time. Right. I mean, this is, again, you're going to get the maximum benefit if you understand your body, and you understand the patient needs to have to play a role in understanding some of their own physiology to maximize this therapeutic approach.
Yeah, that's fascinating. And you've been working with your colleagues over in Turkey and they've been implementing this. Is that correct? What are their results? Have they found anything? Have they been successful?
Yeah, they've been publishing some results, and I've been participatory in some of those publications, not all of them. They're doing a hodgepodge of combining standard treatments with keto, with hypothermia. They do a variety of different techniques. They use lower doses of chemo, and I even asked them. I said, "Why are you using any chemo? It's a poison, right?" So they said, "Well, we have to. They'd close our clinic down if they don't do something."
Standard of care. Right?
No, standard of care, right? Standard of Standard of death is more like it. So, you don't poison and irradiate people to make them healthy. I mean, right? You just don't, you shouldn't do things like this. Yet people do it because they don't understand how you can do something, how you can get the result doing something different. But, and nevertheless, they use lower doses. They use insulin potentiation, hypothermia, and a variety of things. And we published a paper not long ago on the breast cancer page. It's a fascinating story. A woman with triple negative, well, it metastasized to brain, a general metastatic problem, brutalized by the system, from Ohio, this woman. You can read about her. It's published, was it Curious Journal? One of these journals, I can't remember. But anyway, I was shocked when I was reading the case report because the PET scan looked like she was lit up like a Christmas tree. I mean, this was really a devastating, large metastatic spread throughout her body. She almost died on the plane ride over to Turkey, and she was in the intensive care there for a month or so, just trying to keep her alive, I guess. And then when she finally recovered, they put her in the metabolic therapy, started to do remarkably well, came back to Ohio, and the last I got was a picture of her and her husband in Hawaii. And they said I don't remember what hospital it was in Ohio, but they thought that she would have less than a month to live, based on all or the fact that she failed all these treatments, and the cancer was spreading wildly, and yet now here she is alive. Now it's not clear how. I mean, if she's cured, I don't know. I mean, if she dies at 90 years old from like heart failure, then she must have been cured from her cancer. But you can't know if anyone's ever cured until they die of old age from something other than cancer.
Right.
Right? I mean, that's the bottom line. Yeah. So, our man Pablo Kelly was diagnosed with terminal brain cancer. And now he's just about 8 years out. And I said, at what point do we consider the cancer not terminal, right? So, glioblastoma, oh yeah, most people say, oh, it's a terminal cancer. Well, he's still going. He has a website telling everybody what he's doing, how he's doing. And he says, I was diagnosed with terminal brain. Well, yeah, eventually we're all terminal, right? We're all going to die of something. So, so I don't know if we should be using such terms like this in light of the new evidence that we can keep people alive far longer than what was previously thought not to be possible.
And so in Pablo's case, did he end up just essentially switching to a ketogenic diet instead of undergoing, radiation and chemo, and then debulked? Is that what happened with him?
Yeah.
Okay.
So he contacted me right after his diagnosis. I mean, he had been suffering from different kinds of seizures and things until they finally did a PET scan and found out a large lesion in his brain. They took a biopsy of him. That was back in 2014, August 2014. And classified it as a glioblastoma. And then he has a story. He's on various podcasts and things. He gave a blow-by-blow description of what they did to this guy. But he, bottom line, he rejected. He didn't want radiation. He didn't take any steroids. He didn't take any temozolomide. He didn't take anything. But he was he threw himself into this ketosis, this ketotic state very aggressively at the beginning. Also, he had the IDH1 mutation, which is a therapeutic mutation. It's like a drug that targets glycolysis and glutaminolysis. Can you believe this? So, so he has this very therapeutic mutation. He's younger. He avoids radiation and chemo that will accelerate and make the microenvironment acidified and actually contribute to the rapid recurrence of the tumor. So he avoided all. He had certain advantages, and he avoided other things that would have put him at a demise. But then the tumor did grow. It never disappeared. And then in 2017, we looked at the scans. My colleagues, I don't profess to know anything about, all I can see is an image on the scan. But the guys who look at these closely can see small changes, and they noticed that the tumor was continuing to grow. And so they did the debulking. His first debulking was 3 years post-diagnosis with the glioblastoma. So he was keeping it growing very, very slow, using basically a pressed pulse, but he wasn't using any drugs that he could have used that we now know would have been far more effective. But still, They debulked it again, confirmed the diagnosis of IDH1 positive glioblastoma. And then he transitioned back onto the metabolic therapy, and he's doing really well. Then 2021, just last year, he had a second debulking, still the same. So what it is, it's an indolent glioblastoma. It's not growing very fast.
Right. That's fascinating. And normally GBMs, like you said, are terminal. I think their life expectancy is in about 1 year or so.
Well, for his age group, it could be 2 years.
Yeah.
But the fact that he's out almost 8 years now. And as a matter of fact, I just attended the IAOMT meeting, oral toxicology. Pablo now feels that some of his headaches are coming from the mercury in his teeth. So I went to this meeting where they have all these specialists who remove mercury from your teeth, right? So I got him in touch with some physician who does this. But I said to Pablo, I said, you're the first guy on the planet with a glioblastoma that really has to worry about mercury in his teeth.
That's a good problem to have.
Most people don't live long enough to worry about the mercury.
That's right. Teeth. I think it is important to note for the people in the audience that the ketogenic diet isn't necessarily going to shrink the tumor. It might slow down the growth of the tumor, because it sounds like tumor can still use glutamine. And so there's these two pathways, right? I mean, not just glucose. And so there's still glutamine that it can use. Yeah. So do you know, on histology for Pablo's case, was there any necrosis from just being on ketogenic diet? Was that tumor death?
There was some necrosis. You can look at the path reports. First, I didn't believe. When I got the images from his pathologist, Dr. Shivani in England, I said, well, that's Shivani's opinion. That's, because these pathologists also all sometimes have different opinions about what something might be. So I took the slides and I sent them to my colleagues at UPenn, and had the neuropathologist look at it and say, yeah, this is definitely GBM. So I still wasn't convinced. So I had the slides sent to my colleague, Joe Maroon, former president of the Neurosurgical Society of America. And had his pathologist there. They all confirmed. So I had 3 groups of people, because what happens when you get a long-term survivor? They say, he never had GBM in the first place, right? That's the first thing they say. He never had it, right? So, well, I made sure that he did have it, and right. And there was no ambiguity. They all said that this is an invasive GBM. It didn't have as much hemorrhagic it didn't have as much angiogenesis,
proliferation. Yeah.
Yeah. But it was IDH. It was not methylated. It was IDH. So I say to myself, what the hell is this IDH1? So, it's it brings up 2-hydroxyglutarate and all this. So, but people with IDH1 mutations generally have their life expectancy is twice as long as those who don't have the wild type allele. So what's going on with that? Why people with IDH1 mutations live longer than people who don't have the IDH, they have the wild type. So now papers are coming out, showing, if you look at where does the 2-hydroxyglutarate come? It comes from alpha-ketoglutarate. Well, alpha-ketoglutarate is an upstream precursor for substrate level phosphorylation in the mitochondria. So you're going to actually reduce mitochondrial substrate level phosphorylation because you're diverting the upstream substrate into 2-hydroxyglutarate. Not only that, 2-hydroxyglutarate destabilizes HIF. HIF is driving the glycolytic pathway. I said, are you kidding me? I says, this mutation is acting like a drug that's simultaneously targeting glycolysis and glutaminolysis, right?
Right.
So you can kind of neutralize the effects of that drug by irradiating patients' brains, freeing up a lot more glutamine to drive the tumor. I says, when you start to realize the biology and biochemistry of what these cancers are and what we're doing to treat them, in the future, they're going to say, how in the hell could we be so goddamn stupid to do the stuff that we were doing to these people? So it's just going to take time for people to understand what we're talking about. And I don't know how long that's going to take. But, I look at IDH1 as a therapeutic mutation. People live twice as long if you have a GBM, and the biochemical mechanisms are clear. So why they have drug companies now trying to develop drugs to target and remove 2-hydroxyglutarate? That doesn't make any sense, does it? If that 2-hydroxyglutarate is participatory in the therapeutic benefit, then why would you want to take it away? See, this is the thing. People do this, So.
Yeah. Well, that was entirely extremely fascinating. We're coming up on the hour. I want to leave some time for a Q&A here. But before I ask my final question, where can people find you or support your research?
Well, we generate a lot of support from Travis Foundation's Foundation for Metabolic Therapies. Travis has been very good. We're getting support from a lot of family foundations, from a number of family foundations. People make donations to the university. People make donations to Travis Christofferson's foundation. And, this allows us to do what we really want to do without restrictions. And we get support from the English UK Foundation, Childhood Cancer Foundations. The bottom line is, if people are really, really interested in managing the disease and keeping people alive longer with a higher quality of life and longer overall survival, this is what's going to do it. This strategy is what will work for the majority of patients. How do I know this? Because I'm in contact with the physicians and clinics that are doing this, and I'm in contact with patients that are doing it and feeding me back information. So I know this strategy is going to work. It's going to work better than anything that we have in the clinics today, once people understand how to do this. So we work out all the preclinical systems in the lab here. We have the best animal models for glioblastoma. We have a knowledge, we have the know-how, we do doses, timing, and scheduling. We're building drug diet cocktails. And as soon as they're built, you want to talk bench to bedside. This is exactly what we do, bench to bedside. And we publish the papers when the patient lives long enough. I didn't want to publish Pablo's paper until I was sure he was living beyond 5 years. And we have 5 years of data, over 5 years of data on this guy. Right. So these are educational, important things if people really want to look into it.
Yeah, absolutely. And so for my final question, if you had a magic genie and you could wish for the entire world to adopt one lifestyle modification that would most greatly impact their health, what would it be?
Well, I think it would probably be restriction of carbohydrates. I think that's having a massive impact, not only on cancer, because we did not evolve as a species to be eating large amounts of highly processed carbohydrates in our diet. And I think it's not just cancer. I think it's Alzheimer's disease, type 2 diabetes, cardiovascular disease. They're all kind of linked to problems in diet and lifestyle. And, and it's hard because we live in an environment where the foods and the stuff tastes so good. And we're sitting behind computers all day long. We're not exercising as much as we used to, sitting in traffic. I mean, you put it all together, bad food, bad lifestyle, bad issues, and you're going to end up with either cancer, dementia, type 2 diabetes, heart disease, you name it. They're all these chronic diseases. So, but I think if people are aware and they understand the mechanisms, then it becomes a choice, a personal choice.
Absolutely. I love that. Awesome. Professor Seyfried, thank you so much for being so generous with your time. And we're going to switch over to the Q&A. Thanks so much.
Thanks.
All right. That was fantastic. Thanks so much. You have a lot of content out there on the web spaces. And so I was trying to consume as much of it as possible. And you're so extremely generous with, providing your time to provide this knowledge to all of us. So if anybody has a question for Professor Seyfried, go ahead and unmute and ask away. I know that there's probably some questions in the chat, but Galia, welcome.
Hormonal replacement therapy. Yeah, I don't know. I think Susan Wadia Els just published a book that dealt with a lot of these hormonal issues, and I think she cited reference papers that address that more. She was talking about birth control pills and certain other hormone replacements and things like this. And I can't be sure what's going on with that. I don't know if you provoke cancer, you stop cancer. I mean, I don't know. I really can't answer that question.
Out of curiosity, with the press pulse, does that work with every type of tumor? I know you talked about GBM and breast cancer, but does it work for others as well?
Well, you know, I in our big iScience paper, I went through all the major cancers, and I looked at the electron microscopy evaluations of all the major cancers in human tissues, and all of them had, and biochemistry analysis of mitochondrial proteins and enzymes, and every one of the major cancers had defects in the mitochondria. So that would mean that everyone had to ferment, and we all know that, you know, glycolysis is big in almost all the major cancers, not all of them, but most of them. So they're either using glucose or glutamine. Other than that, they can't use anything else in any great degree. So yes, all these cancers are pretty much similar in their underlying biochemical problem. So that means a singular solution, a singular approach would be able to manage the majority of cancers.
Yeah. Discussing the structure of mitochondria, I know that I think it was like Lou Cantley's group had published something back, you know, a while ago now, but it was in Science. And they were talking about that it potentially that the Warburg effect wasn't necessarily due to dysfunctional mitochondria. I didn't quite get the punchline. I was trying to read up on it, to be honest, right before this. Do you remember that paper? Are you familiar with that?
Yeah. Well, I don't know how, if every cancer I looked at, if every of the major cancers that have been evaluated have clear structural defects in the mitochondria. Do we deny that, or do we ignore it? How could you say cancer doesn't have a mitochondrial defect when you're looking at the tissue showing it's mitochondria defective in the tissue that you're looking at?
Right.
So I don't know, and I also know a lot of people just ignore those facts.
Yeah.
You know, and you know, Albert Leninger wrote the book. In fact, I have it here. Structure determines mitochondrial function.
Mm-hmm.
And if you have structural defects, everybody knows if you have a structural defect in the DNA, you get a mutation that can sometimes have a problem. So if you structurally alter your mitochondrial cristae, mitochondrial associated membranes, are you assuming that has zero effect on the ability of that organelle to perform normally?
Right.
So again, this comes back to evolutionary biology. I mean, if the function of that, or if the function is dependent on the structure, and the structure is abnormal, how do you say that mitochondria are not defective in these tumor cells?
Right.
Right.
Yeah. Yeah. No, it seems to make sense to me.
Yeah. Well, you have to look at that's a lot of people. They don't look at the actual EM images. They don't have time to sit and start looking at all these. Go look at all the EM images, and you're going to say, "Oh my God, look at these mitochondria. They don't look normal, do they?" So it's just black and white. I don't know. I mean, as I said, what I find is most people don't study the literature. They base their information on hearsay and what others have said.
Yeah, absolutely. Do you have time for a couple more questions, or?
Yeah, sure.
Does anybody have a question? I've got plenty, so I just want to leave time for.
Fire away. In fact, I love the questions. Sometimes I find somebody asks a question that I really need to think more about, and then I'll spend time going back into the literature to find the answer. Yeah. But you know, that's the only way we can move the field forward. If you're able to provide an intelligent answer to any kind of a question, then that illustrates that you might know what you're talking about. Right. And if you don't know the answer, you know, I don't want to bullshit about it, but you know, bottom line is that I either know or I don't know.
No, I love that. Is there, so we talked about the caloric restriction ketogenic diet as part of a therapy once you already have cancer. And I know that caloric restriction has been shown to potentially slow down tumor growth, but is it also for prevention as well? Is there anything that has indicated that it would prevent cancer?
Yeah, sure. Well, first of all, you have to say, what is caloric, you know, caloric restriction in a mouse is not the same as calorie restriction in human. We published a big paper on this. And we found that a 40% calorie restriction in the mouse, which gives you really, really good effects on most cancers, is comparable to water-only fasting in humans. So and as a species, we evolved to fast. Humans can fast for long periods of time because we had to do that to survive in the environments that we transitioned through during our evolutionary past. So if the origin of cancer is damage to the oxidative phosphorylation and structure and function of the mitochondria, then if you keep your mitochondria healthy, it's very unlikely that you're going to get cancer. So how do you keep your mitochondria healthy? By burning ketones. Ketones will reduce the production of reactive oxygen species. So we all die from entropy, disorder. So, and the order in our bodies is maintained by keeping us away from equilibrium due to the energy efficiency of our mitochondria. So the better you can keep your mitochondria healthy, the less likely you are to develop the diseases that are linked to mitochondrial dysfunction.
Right.
So yes, it's a powerful, and you know, our closest relative, the gorilla or the chimpanzee, and I wrote about this, they rarely, if ever, get cancer. They've had a documented case of breast cancer in a female chimpanzee, and yet they're 98% similar to us in gene and protein sequences. So what the hell is going on with the chimps? The problem is they're eating the same, they're having the same diet and lifestyle they had when they first evolved as a species. We as a species are not in the same diet and lifestyle that we had when we evolved.
But bananas aren't ketogenic though, right?
Well, they don't eat a whole buttload of them all day long. You know, I mean, it's like a treat, right? You don't just throw in a whole branch of bananas and that's your meal for the day. You know, it's, you know, they're eating a variety of things, and they're eating many of the same. Of course, they kill monkeys and eat raw monkey meat in the jungle as an occasional. They eat insects and they eat these kinds of things, but they don't get they don't get cancer. And it's if you look at the foods they eat, they have these powerful mitochondrial protectors in the foods that they're eating. So I was to the zoo down here in the Franklin Park Zoo.
Yeah, I was just there.
Yeah. You see the gorillas. So I talked to the vet in there, and I said, "Yo, what are you feeding?" "Oh, we feed them their natural diets." Right? Go ask the veterinarian. So I said, "Why don't you give them a McDonald's hamburgers and a big pizza every now and then?" "Oh no," she said, "that would be animal cruelty." She said they would get sick.
Yeah, I bet, especially if you're not used to eating that sort of thing.
But it wouldn't take them long to get used to it.
No.
You put a couple of big jelly-filled donuts in there and see what their lifestyle would be.
Yeah. Actually, Heidi mentioned something about dogs getting cancer, and I saw one of your presentations where one of the somebody that you knew their dog had this some sort of nasal pharyngeal carcinoma of some sort, and switched to a ketogenic diet, and it like shrunk on its own without any radiation.
Oh yeah, it happens a lot.
Yeah,
the dogs.
That's fascinating.
You know, the dogs are loaded with cancer too because of we're feeding them the same crap that we eat. You know, all the dog foods are filled with grains and corn and all kinds of crap. The wolf, they all came from the wolf. When was the last time you saw a pack of wolves attack a cornfield? They don't eat corn, right? They kill chickens and other animals. So if you feed any animal the wrong diet and keep them in a cage, they're going to get sick. No exercise, eating the bad food. I mean, it's very clear, right? So
yeah,
it all comes down to the same thing.
Speaking of dogs, Stephanie has a question, but she actually does dog agility competitions. So let's go to Stephanie, who has a question.
Yeah. 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 That's level 2, I think. I think people come to realize that the more the fact that they're still alive and saying, wow, I'm doing better. I feel much better. Let me switch now to some more organic keto and see how I do. And I think that's part that will become an extremely important part of the overall solution as you as you start to see benefit, the benefit could be improved more with recognition of those kinds of foods that are even more healthy, but yet can sustain the therapeutic ketosis. So I think that's absolutely going. We're still so much at the beginning here. We haven't really explored the value of what you just suggested, and I think that's going to be an important component as we move forward.
Yeah. Steph, anecdotally, I started pretty much organic, grass-fed, et cetera, and I feel much better. I feel like I can eat more of this type of food and feel great, whereas before I'd feel a little bit sluggish, and I've felt even better since switching to keto. I actually switched to keto the day that Dom said he was going to come on the show, because that would be disingenuous to have never tried keto, but to have Dom on. So I went keto essentially that day. I fasted 24 hours, switched over to keto, and felt pretty great. So I think I'm going to keep it up for a few more months here and go from there. Max, did you have a question?
Yeah, well, that's that I mentioned was the oncogenic paradox, which could be any number of things in the environment. They're called risk factors. They're cancer risk factors. So some people can be exposed to a carcinogen in their workplace or even in their home. We call these chemicals carcinogens. And what I have found in my research, that the carcinogens often cause a natural fluorescence within the mitochondria. So the mitochondria are actually being damaged by carcinogens. You can have intermittent hypoxia, which is known to damage mitochondria. Chronic inflammation in the body, like if your C-reactive protein is elevated, that those inflammatory cytokines that can damage mitochondria in a particular cell of a particular organ in some people. Therefore, the cancer could arise from systemic inflammation. You can have some people get cancer from various viral infections like hepatitis C, papillomavirus. These are called oncogenic viruses because either the virus themselves or the product of the virus damage the oxidative phosphorylation. And it's never acute, it's always chronic. Because as Warburg said, anything that would cause acute mitochondrial damage would lead to cell death instantly because of the apoptotic signaling cascades. But what happens is, as the mitochondria gradually over time, protracted loss of their ability to generate oxphos, oncogenes upregulate fermentation mechanisms. So making HIF and these things, they're all upregulating transporters for glucose and glutamine and things like this. And eventually the mitochondria lose their ability to regulate apoptosis. So now you have a renegade cell, a cell out of control. So, but it can happen from a number of different things. Even the BRCA1, these breast cancer genes, and Li-Fraumeni and some of these other, they damage mitochondria, put the cell at risk for forming a tumor. So, because we have not yet found a mutation that's 100% penetrant, can we say that that mutation is absolutely caused? Most mutations, inherited germline mutations, are risk factors. Some of them can be very good risk factors, like Li-Fraumeni, and others very weak risk factors. So, but they can still, in the right environment with that risk factor, you can push the cell into a fermentation metabolism over time. So, yeah, there's a lot of things that can damage mitochondria over in a protracted way. Yeah, yeah.
You know, uh, yeah.
Yeah, it comes through what remnants of an electron transport chain might be there. So the ROS are coming predominant, but they also come from other parts of. There's other organelles. Some might maybe I can't remember it right now. It slipped my mind. But there's other places where ROS can come, but the majority come from the mitochondria. and especially under these different kinds of hyperoxia, hypoxia, these kinds of things, they produce these RAS, which then further damage mitochondria, and then at the same time can cause mutations in the DNA. Yeah, well, RAS are reactive species, so they'll actually cause nucleotide damage. They can actually cause the damage to the lipid membranes are very important, especially in the mitochondria, where you have a lot of polyunsaturated fatty acids in the inner membrane. And the cardiolipin is the major lipid of the inner membrane of the mitochondria. And we published major papers showing how the cardiolipin is all screwed up in these inner membranes, which regulate the function of the electron transport chain. So multiple systems within the organelle itself can lead to the production of RAS, with further damaging the system, leading to a compensatory fermentation driven by oncogenes. They are transcription factors that upregulate fermentation metabolism. So they are facilitators of the transition from normalcy to malignancy.
Right. Professor Seyfried, I want to be respectful of your time. Do you have time for, let's say, three more questions?
I do.
Okay. Heidi wants to know, how compatible is the research results with rats versus humans?
Mice, predominantly. I don't work with rats. I work with mice. So the rodent, how compatible is the mouse to the human? Extremely important question. In fact, this is what the nice thing about the mouse is that you can get your results much faster than you can in the human. The human responds better than the mouse to the treatments. This is what we always find remarkable as well. And the difference, why that's important, is because the basal metabolic rate of the mouse is 7 times faster than that of the human. In other words, the human is 7 times slower than that of the mouse. So when we study human cells in culture, we have to expand how long we need to watch these cells, because their metabolism is a lot slower than that of the mouse. We can see stuff in the mouse real quick, but we have to wait a lot longer in the human. The mouse cannot transition to ketosis as well as the human can. Humans are much, much better at transitioning to ketosis than the mouse is. So the therapeutic benefits of our studies in mice, we always have to keep that in mind. The differences in basal metabolic rate. So when we start to use dosage, timing, and scheduling strategies in the clinic, we have to always be cognizant of the fact of the differences that we see. How we translate dosage, timing, and scheduling from our studies in the mouse have to be modified in certain ways due to the differences in basal metabolic rate. And that's also overlooked in many of the experiments on xenograft models and patient-derived xenografts and all these kinds of model systems, where you put cells from one organism into the body of another organism. That's 50 million years of separation and evolution. And you don't necessarily see the same kinds of responses that you would expect in the natural state of these cells. So this is an extremely important point that a lot of people don't consider, either in practice or in design of experiments.
That's a great answer. I had a question when you were talking to Max, answering his questions. So let's say you have an HPV-driven, cervical cancer, and you mentioned that the virus is causing damage to the mitochondria. And I was wondering if there's anybody that has looked at the same cancer, let's say driven by HPV for the purposes of this cause, and if they've looked at EM and looked at the structure of the mitochondria, is it the same every single time because the source was the virus? Is it the same damage?
Yeah, no, that's a great, I don't think anybody has done that. Because, when you get a cancer, a lot of the people doing the EM work, the pathology, never addressed, how did this person get their tumor? How are we seeing the pathology in this person versus the pathology in another person that may have had a different origin? All we're looking at is the end result of the insult, which is this dysregulated cell growth in a population of cells in a particular organ. Mm-hmm. I think that kind of a question becomes relevant now that you understand what the origin of the disease is. So I think that would be a really interesting strategy to look at now that you become aware of where, okay, because they would say, oh, this person has ovarian cancer, or this person has breast cancer, and they, well, some of them are one kind of, and they switch, right? Somebody can have a HER2 negative breast that develops into a triple negative. What's going on with all that? And so those kinds of questions, I think, can be maybe addressed now that we have a better understanding of the origin.
Right. Yeah, fascinating. And then the last question that I had, again, I was thinking about when you were talking to Max, again, if things are derived due to mitochondrial damage, yet we do see certain genetic mutations that are common themes amongst many cancer lines. So what is it that is, what's the signaling thing that from the damage of the mitochondria that leads to, let's say, something that's common, like a KRAS mutation? Why is it selectively causing that particular damage within the DNA?
Yeah. Well, it might have to do with the structure of the gene itself. Like a lot of cancers have mutations in P53.
Right.
And I question this because I said, but they don't all. As a matter of fact, we know from doing genomic analysis of individual cells and tumors that there's no two cells, no single cell has the same genetic abnormalities as any other cell in the tumor. So there's a lot of heterogeneity, but yet every one of those cells is fermenting, regardless of what the types of mutations that happen to be in that particular cell. So let's just consider the P53 gene, because that seems to be more common than, say, some of the other genes.
Sure.
Okay. And when I was reading at this at the beginning, it was supposed to be the guardian of the genome. They would call P53. It codes for cytochrome C, one of the cytochromes in the electron transport chain, which then can link it to that. And then there's some people who have P53 germline mutations that don't develop cancer.
Right.
They're not always. You only get the cancer when you actually damage the mitochondria. But why the P53 or some of the other genes that you see more commonly associated with mutations? It may have to be the vulnerability of the structure of that gene to reactive oxygen damage. So, and then when when they said P53 was the guardian of the genome, well, cells that are important, a gene that would be really, really important in evolution to keep ourselves functional, should have very rare mutation, not common mutation. Like, for example, histones very rarely have, they don't tolerate genetic variation at all. They're very highly conserved, even with yeast in humans. I mean, so whatever they do, it's so essential for the function that you cannot tolerate variability, heterogeneity in the structure of that particular protein. Same with some of the proteins of the electron transport chain. They're highly conserved across species because they can't tolerate heterogeneity and still function optimally. Now, why the guardian of the genome would be so prone to genetic damage makes no sense. Because if you're the guardian of the precious genome, how is it possible that this gene can be mutated more than most of the other genes? And it might have something to do with the structure of the gene being vulnerable to reactive oxygen species. And there was a paper published in PNAS that actually looked into this. The P53 is extremely vulnerable to ROS damage. So what's his name? Published a couple of years ago. I have it cited in one of my papers.
Yeah.
But he looked into something like that. And so you can think about those guys who love to study the intimate structure of these genes. For me, I don't worry about that because the damn cell is fermenting. So if I pull the plug, I don't have to worry about whatever kind of mutation is largely irrelevant. Right. So they can't live without those fuels, so you can kill them. So why worry about what mutation they have?
Right.
But you're right. That's a good question, and I think it has to do with the vulnerability of. That's one explanation. There could be others.
Sure. Actually, just when you said that, I had one last question. Sorry. Thank you so much for your time. But is there any potential that, let's say, the press pulse, it gets widely adopted? Is there any chance of resistance, a tumor cell growing resistant to a press pulse protocol?
Unless everything comes down to ATP. So if you can make ATP, you can survive. And the question is, how are you going to make it? So if the mitochondria become normal, then the cancer will, it won't be a tumor. Right. In fact, mitochondrial medicine is being considered as a possible therapy now. Replace bad mitochondria with good mitochondria.
Interesting.
And that's basically the essence of the nuclear mitochondrial transfer experiment. Right. So if you can replace bad mitochondria in your tumor, you're not going to have a tumor. So the tumor will be managed. But is there a way that a cancer cell could eventually become resistant to a press pulse therapy? Only if it can have access to glutamine to generate some sort of energy. Because without everybody, you have to realize everything is downstream of ATP. Because without energy, nothing can grow, nothing can survive. Everything is downstream of energy. So glycolysis, that is down. So you got to have energy to do something. So if the cancer cell is dependent on fermentation for energy, then targeting that, how can a cancer cell survive without energy? And unless they say, well, I can generate energy from asparagine. Okay, how much asparagine is in the microenvironment that's going to allow that tumor to grow out of control? Because there is a little bit of boost from asparagine but never like glutamine. Glutamine is the big dog. All the other some amino acids are actually inhibitory, like methionine. So you'd have to say, how is that cancer going to get any fuel that's going to replace glutamine and glucose for energy? And then you'd have to think about that. And there may be certain circumstances that could do that, but I'm not aware of them.
Right. Fascinating. This was amazing. Thank you so much for your time and being so generous and coming and educating us. I find this extremely fascinating. So thank you so much.
If anybody has any, feel free to email me. I'm always happy to answer questions, because the more questions that I can answer effectively, the more likely I'm right.
That's true.
Right? I always tell my students in the class, prove me wrong. Prove the things that I'm telling you, prove me that I'm wrong. Oh, this paper over here. So let's look at that paper and go through it, dissect it deeply. And we find there's flaws in there. They didn't do the controlled experiments. They overlooked something else, or they assumed something that wasn't true. So that's the way it is.
Yeah.
But I know in the future we will be doing press pulse metabolic therapy, and cancer will be recognized as a metabolic disease. They just don't know how long it's going to take for the field to come to realize this. That's
right. Well, I love that,
right.
Professor Seyfried. Thank you so much.
Yeah,
Really
thank
appreciate
you very
your time.
much. Yeah.
All right.
Bye now.
Bye.